So as we continue with these reaction mechanisms, I'll probably begin going through the reaction mechanism fairly quickly, and then we'll kind of return to it to kind of talk about some of the subtleties of it and some of the potential applications. So this one is the first one where we maybe have a little bit of a question about, well, how do we write our starting material? So again, with this one, I just want you to realize that we're generating a good leaving group here. So I'm going to draw out what the structure of this is, but I indicated for your mechanism that you can basically just write an OAMP group. So I'm going to draw out what this looks like, and then we'll abbreviate it. So again, this whole group right here, we are just calling OAMP. So this is adenosine monophosphate. So we'll learn more about the structures of these when we have our nucleic acids chapter. But again, the whole purpose of this is that we generate something that is a good leaving group. And so that's why we have this. Like in our second reaction mechanism, we had something that was a good leaving group and a carboxylate group. But again, that was a way to chemically activate. This is how biological activation occurs. So we're going to react this with our amine. So again, our nitrogen is still going to be our nucleophile. And now we're going to have that same carbonyl carbon as our electrophile, generate our tetrahedral intermediate. Most of the reaction mechanisms we're doing here are going to be reversible. So generating our tetrahedral intermediate, and I'm now going to abbreviate this just as our OAMP group. We're going to see a very similar mechanism to what we had with reaction mechanism 2. We're going to generate a tetrahedral intermediate where we're going to need to do some proton shuffling in order to get the correct protonation states. So again, we've got this tetrahedral intermediate where our carbonyl oxygen is now negatively charged, our amine nitrogen is positively charged. So again, you can always use water as a solvent, or as we saw in our previous mechanism, we can start thinking about using either A- or HA, or B- or BH+ is what I tend to use. And again, water is always good. But what we need to do is we need to do some proton shuffling. So we're going to protonate this guy, deprotonate this guy, and this leaves us with our final carbonylamine intermediate. And the purpose of doing this reshuffling is that when we want to decompose this tetrahedral intermediate, we are going to break the sigma bond to our best leaving group, our weakest base. And if we didn't do this proton shuffling, then the weakest base would have still been that protonated amine. And so this allows us to then decompose our tetrahedral intermediate. We're going to displace those sigma electrons onto our good leaving group, generating our protonated carbonyl compound, our amide, and then I'm going to redraw out at the end what this looks like, but our OAMP good leaving group. Okay, the last step is that we need to get rid of, and I'll just keep water throughout this entire mechanism as either our acid or our base. So we're going to have water is going to effectively deprotonate that guy, generating our amide. So I'm just going to kind of redraw out what this guy looks like. And I'm just indicating the nitrogenous base adenine as an A there. But that's what that guy looks like. And just to highlight again why this guy is a good leaving group. So again, good leaving groups are weak or stable conjugate bases. So something that's going to be a conjugate base is usually going to be something that's negatively charged, and that negative charge is sufficiently stabilized such that it's not trying to find a proton to get rid of that sort of unstable situation. Remember, we talked about chemistry always happening so that things become more stable, go downhill in energy. And if you have something that's negatively charged, potentially, a basic molecule, but you've got resonance stabilization, you've got inductive electron withdrawal, you've got all kinds of ways that you're stabilizing these negative charges, then we have a situation where that's not really a strong base. And so it's able to move away with those sigma electrons and sufficiently stable them as an anionic species. So building on what we learned in reaction mechanism 2, we see another situation where carboxylic acids can do chemistry with amines and do form amides. But basically what we need to think about is how we generate a leaving group that allows us to do chemistry with that carbonyl and importantly get rid of that acidic proton, which was a better electrophile. So what we see here in reaction mechanism 3 actually is how proteins are made. So a very common biological leaving group we're going to see here, it's a theme that we'll see throughout the course here, is a nucleotide either monophosphate or diphosphate group. So again, in this case, we're considering adenosine monophosphate, again, representing it generically as OAMP. We haven't talked about the structures of nucleic acids, and truthfully, if we flip back, there was nothing about what was in that AMP group that really made it be a good leaving group. We just needed to know that it was a good leaving group so that it could easily depart. And again, this chemistry occurring in slightly alkaline conditions, physiological pH being 7.4, you might not consider protonating the carbonyl oxygen. There's a supplemental sheet that I'll make available that talks about whether or not you protonate. It's called to protonate or not. That is the question. It's basically a pKa argument. So you wouldn't lose points if you did not show that you protonated this carbanolamine if you didn't show protonation at this point. That would be okay. You do need to show deprotonation of the protonated amine though, because otherwise that's still the best leaving group. So we'll start now as we're learning about reaction mechanisms, now that we start having ones that we're going to see biological homes for, talking about a biochemical application. So this is a part that you're going to want to include on your reaction mechanism, just kind of highlighting why this is important. So again, the biochemical application for here is that this is how we do form amide bonds in biochemistry. So when we polymerize amino acids, we're going to connect those amino acids via peptide bonds to form proteins. We're not going to study the details of that enzymatically in this course, but we will when we consider protein structure, look at the structure of a peptide bond, and we'll revisit this mechanism to kind of remember how that activation occurs.